Hi, I'm Josh. This is our Enzyme Lab Presentation. Trypsin is a protein that acts as an enzyme in the small intestines of mammals. It's a catalyst for breaking down proteins in the mammal's diet. Enzymes speed up chemical reactions by lowering the activation energy needed for a reaction to proceed. Enzymes are important in living organisms because they can help speed up the breakdown of substances in the animal's body. They also regulate reactions by making them occur quicker and more frequently. Trypsin cleaves apart peptide bonds on the carboxyl side, mainly after amino acids like arginine and lysine. Its role in the intestines of an animal is to break down proteins for digestion. Our question we want to answer in this enzyme lab is, at what temperature is the activity of trypsin at its peak? Our hypothesis is that trypsin will peak around 57 degrees Celsius, and in the temperatures leading up to 57, the activity will gradually increase. However, beyond that, it will plateau before dropping off at 97 degrees Celsius. Our rationale behind this is, as the temperature increases, the increasing molecular movement will increase trypsin's contact with peptide bonds. However, the temperature gets to the extreme end, it will denature the enzyme. Materials and methods. We took 42 centrifuge tubes and labeled 21 as the control group and 21 as the experimental group. Doing so, we had 3 tubes for each of the 7 temperatures we tested, and 3 tubes for each control at every temperature. To the experimental tubes, we put 500 UL of azocasein and 50 UL of trypsin, and 450 UL of a pH of 9 NaCl buffer to the experimental group. To the control group, we did the same, but instead of adding the trypsin, we substituted it for water. Then we incubated each centrifuge in 37, 47, 57, 67, 77, 87, and 97 degrees Celsius for 20 minutes. After that, we added TCA in order to denature the trypsin and combine with the undigested substrate. Then we centrifuged for 2 minutes on max setting to separate the pellet and the supernatant. After the centrifuge, we used the spectrometer to gauge the absorbance of the solution at 440 nm. Negative controls and replicates. To account for any substrate breakdown that occurs even without the trypsin, for every temperature, we included 3 negative controls, which we prepared the same as the experimental, except that we substituted trypsin for water. The negative control lets us know what amount of azocasein breakdown should be expected even without the enzyme. Another method that was utilized to protect us from variances in our results was that we made sure to have 3 replicates for each experimental and control group at each temperature. That way, if we have one measurement that is way apart from the other 2, we can average it out or not include it, protecting us from making conclusions that are false due to mistake in our data. Enzyme assay. After the trypsin was given its time to hydrolyze peptide bonds, trichloroacetic acid, TCA, was added in order to denature the trypsin and precipitate the denatured trypsin with the undigested substrate. We then centrifuged it so that the more dense pellet of denatured trypsin and undigested azocasein formed a pellet at the bottom, while all the shorter digested azocasein collected at the top, suspended in solution as the supernatant. We tested how different temperatures affected the enzyme, and for our controls, we had the pH of the solution set to 9. We did this because, seen in other groups' labs, 9 seemed to be the ideal temperature for pH for trypsin activity. We also made sure to keep a consistent amount of TCA, trypsin, and azocasein so that the results do not become skewed due to concentration differences rather than the temperature. Substrate. Azocasein, a chemically modified protein prepared by adding sulfamylinide groups and covalently linking them to the peptide bonds. When the peptide bonds are cleaved, this allows the sulfamylinide groups to be released as well, and these groups can be detected by absorbance at 440 nm, thus allowing a measurement of how much the substrate is broken down by the enzyme. Here's the results of our data. As it is seen here, the variability of our results were extremely high for most data points. In particular, the variability between control and experimental for 77 and 87 overlap. The average trypsin activity at 37 degrees was statistically different from 47, which was statistically different from average activity at 57. However, the activity at 57 was not statistically different from 67 since the variability graphs overlapped, nor was 67, 77, and 87 from one another. As seen from the t-test results, 37, 57, 77, and 7 are all values greater than 0.05, meaning that the difference between the experimental and control group was not statistically significant. What the graph does not show is that the activity from 37 to 47 increased by 0.36 before decreasing by 1.02 to negative 0.31 at 57 degrees. Then it further decreased to negative 0.39 and 67 before increasing to negative 0.22 at 77, then negative 0.15 at 87 before increasing even more back to the positive value of 0.15. Unfortunately, as seen from our p-values, 37, 57, and 77, and 87 failed to reject the null hypothesis. As you can see from our graph right here, we had a lot of standard deviation between the bunch, which may have been due to error on our parts. So some of our data might be slightly skewed. Okay, and finally we're at our discussion and our conclusion to our enzyme lab. The hypothesis for the lab was that enzyme activity would increase as temperature increases until temperature reached around 57 degrees Celsius, where it would then begin to decrease until it reached 97 degrees Celsius, where it would plateau. Enzyme activity would then decrease until hydrogen bonds are broken. From our test, our hypothesis was partially supported. Enzyme activity slowly increased until it reached around 47 degrees Celsius, where it then slowly went down. And then we found that the optimum control group temperature was around 67 degrees Celsius. The experiment was adequate in testing our hypothesis. Our control variable did result in the highest enzyme activity. We used a pH of 9 buffer because groups who tested for pH earlier determined that was the highest enzyme activity that occurred. Overall, I think we did a good job on our enzyme lab. We definitely should have retested our samples. We have a really large standard deviation, and that shouldn't be there. We definitely should have resampled and try to get the data within reason, not so far off. And also, I think part of that comes from our centrifuging. We might have messed up and centrifuged a little bit of the supernatant and centrifuged a little, maybe not enough, because the standard deviation is really high and might have miscalculated some of our data because of that, and also led to the wrong p-values. And basically, from our lab, we concluded that the ideal temperature for trypsin is around 47 degrees Celsius,